Happy Friday, finally. Wednesday seemed like Friday, and now it's really Friday. How's everybody doing? Everybody's starting to think about finals, so I'm starting to get some questions about finals and so forth. So let me answer some of the common ones, one of which I just got over here. That is, what percent of the final will be new material and what percent will be old? And so I make them proportional to the coverage in the class. So there will be roughly 20 to 30% new material, and the rest of it will be things from what you've already seen. Common question, how do I study for the final? And my answer is, how did you study for everything else? But the strategy I would give you is use the exams that you've had as an outline. Okay? They provide a very good outline for what you've seen. You might see a couple questions exactly the same, but most of the questions will not be the same. But they'll cover the same topics. And I'm not going to go back and dig up things that were obscure that I haven't tested you on before. So that's why using those as a guideline for your studying can be helpful, I think. Okay? As I mentioned before, you will get a note card to use on the final. You have to get that card from me. Okay? I will bring that to class. That is a 5x8 note card. You can use both sides. And you have to turn in the card when you're done. So that means that even if you decide you don't want to use a card, you still have to turn in a card. So you need to get a card from me. Okay? If you don't have a card to turn in, you will lose points. So it's important that you get the card from me and that you turn in a card, whether it has something on it or not, with your exam. Does that make sense? I'll pass those out next week. Now, I said I would make myself available today, and I will, after class, for those of you who want to bring in your corrected exams. And I'm emphasizing that people need to be professional in the way you're putting these things together. I've seen some very sloppily put together things so far, so make sure that you're doing these in a professional manner. And you need to meet with me about that. So that's why I'm going to be available after class today. I'll make myself available for quite a while, so I apologize. Some of you are going to have to wait a little while in line to connect with me, but we can pull a lottery or something for deciding who gets to go whenever, although I have promised one student that she could go first because she had been in touch with me about some other stuff already. But short of that, we'll do a lottery or something to get decide the order to get everybody in, okay? I will also, I know on my schedule it's very difficult to connect with me, so if we don't get everybody today, I will promise I will certainly work everybody in who comes over today, even if that means I'm here quite a while. But if there's some of you who can't come today, I will make myself available also on Tuesday evening, okay? So, and we can decide from that, okay? So that's what's up. So today I want to, last time I talked about lipid metabolism, starting off with fats and fatty acids. Today I want to finish up a couple things about the catabolism of fatty acids. And then I want to go through the synthesis of fatty acids, which will be important, ketone metabolism, and then I will talk a little bit more, well, this is ketone metabolism, and then the synthesis of fatty acids down here. So that's the plan for the day. So I put it out last time that fatty acids made it into the mitochondrion where the beta oxidation process occurs on the backs of tiny carnitines, as it said in the song, okay? And that meant that that was necessary for their transport to get into the mitochondrion. I took you through the beta oxidation process, and you can see it's just an iterative process. 2 carbons come off each time, and they come off as acetyl-CoA. And since you've seen how acetyl-CoA itself can be oxidized in the citric acid cycle, it's pretty apparent what happens with that. We have, if we had something that had that had 18 carbons, for example, that would be 9 acetyl-CoAs that would be produced, and that would take 8 cycles because the last one splits 2 in half. Okay, so 8 cycles to basically oxidize 18 carbons. Now, there's not I mentioned briefly that there is an acetyl-CoA dehydrogenase that is comes in 3 forms. That was the one that was deficient in sudden infant death syndrome infants in many cases. I briefly mentioned that one of those was found in the peroxisomes, and so I mentioned that the longer chain fatty acids actually start their oxidation in the peroxisomes. They start getting oxidized in the peroxisomes. I haven't talked about how they get in there, and you're not responsible for knowing how they get in there, but suffice it to say that they do get oxidized inside the peroxisomes. and then they get moved into the mitochondria for the remainder of their oxidation. Now, there are other considerations in oxidation of fatty acids. Stearic acid is a saturated fatty acid. So what I showed you for beta oxidation works beautifully for saturated fatty acids. Okay? It doesn't work quite as simple if we have either odd carbons or if we have unsaturated fatty acids. Because if we have an even number of carbons, then that last cut splits a 4-carbon molecule into 2 acetyl-CoAs, and everybody's happy. But what if that last structure has 3 carbons in it? Well, it turns out that the enzymes won't split that 1 carbon off from the 2, so the cell has to make some unusual adjustments, and I want to show you that very briefly here. Don't worry too much about the structures, but I want to show you something odd that happens in the metabolism of the odd-chain fatty acids. So if I have a fatty acid that, for example, has 17 carbons in it, and yes, our body does occasionally encounter this because some organisms actually have fatty acids that have odd numbers of carbons in them. The beta oxidation process will continue just like it will in the even carbon fatty acids until you get down to that last 3-carbon piece. That last 3-carbon piece is known as propiononyl-CoA, and that's what you see right here on the left. So we've had beta oxidation that has broken off 2 carbon pieces all the way until we got down to this guy right here. And at this guy right here, the cell has to metabolize it in a different way. So, Gesundheit. So let me show you what happens with this. And it's really this is one of the odder things I see or think about metabolism. This guy has 3 carbons, so the cell decides, okay, well, to metabolize this, I've got to make something that has 4 carbons. And the easiest thing for it to do would be to put a carboxyl group on there. And if it put a carboxyl group onto there, you would think it would just simply put it onto the end and it would make succinyl-CoA, because succinyl-CoA can, of course, be oxidized in the citric acid cycle and everything would be fine and dandy. Well, the cell, however, likes to make students study biochemistry, so it puts a few extra steps in there that, again, I'm not going to hold you responsible for, but I'm just kind of showing you this to show you some of the quirks of metabolism. It takes that carbon dioxide and it puts it not on the end, but it puts it on the middle carbon. Okay? And that makes something called methyl malonyl-CoA. And these things here, valine, for example, means valine can be broken down into this. Or some amino acids can be broken down into this, et cetera. That's all that's indicating. It puts it on here, but then it decides after it's put it on there that it put it on the wrong configuration. Instead of having it in the D configuration, it should have had it in the L configuration. So it flips it. Okay? So it's doing 2 steps here that would seem to be ridiculous if all it had to do was this. Because then after it flips it, it takes that carboxyl group and finally puts it onto the end. So we like to think about metabolism as being all simple. Maybe you don't think of it as simple. I think of it as simple. But we think of it as very logical and all, very planned out and so on and so forth. And of course, this is the way that the system evolved. But it's very interesting to see how this came about ultimately to give us succinyl-CoA. So the bottom line of what I'm showing you here is that odd-chain fatty acids yield propiononyl-CoA, and propiononyl-CoA can be ultimately metabolized into succinyl-CoA. Okay? And there's one other thing I want you to retain about this, and that is that there's a very important coenzyme for this process. And that coenzyme is known as vitamin B12. This is one of the reasons that you have to have vitamin B12 in your diet so that you can metabolize fatty acids that have odd numbers of carbons in their chain. There's other reasons you need that too, but this is one of the reasons that you need that. And vitamin B12, you may or may not know, has cobalt in it, and it's the only reason I'm aware of that we have to have cobalt in our diet is because of vitamin B12. Okay, so that's the odd-chain fatty acids. When we get into unsaturated fatty acids, there's a couple of wrinkles that happen with them. Okay? And you might think, well, it's all fine and dandy because in the beta oxidation process, we make an unsaturated intermediate, and everything should be fine and dandy with that, but it turns out it's not quite that simple. The unsaturated intermediate that we make during the beta oxidation process has a trans double bond in it. And as I've mentioned before, I think, that trans double bond is not what gives rise to trans fats and trans fatty acids. Those happen as a result of chemical treatment of food. Okay? This trans intermediate that's there is simply that. It's only an intermediate. It doesn't contribute to trans fat or any of that sort of stuff, but it's an intermediate in that beta oxidation process. When we look at the unsaturated fatty acids that are in our diet or that are in our body, they don't have a trans bond. They have cis bonds. And so that difference in configuration means that they can't be metabolized directly in the beta oxidation process. So what I want to do is show you one simple way in which they can be metabolized, okay? And I'm going to leave the more complicated way out for you. The simple way involves this enzyme right here, enoyl-CoA isomerase, okay? Now, I told you in fatty acid oxidation that everything happened between carbons 2 and 3, right? Everything happened between carbons 2 and 3. So we can imagine we've got a long chain of fatty acids out here, or a long chain of carbons out here in this fatty acid, and they're going to get broken off 2 carbons at a time until we finally get down to a structure that looks something like this, okay? And if we look at this guy, we start counting carbons. We see 1, 2, 3, 4. Okay, and we've got a double bond. Well, where do we want the double bond to be? Between 2 and 3, because all the action occurs between 2 and 3. So it turns out that this is in the wrong place, and it's in the wrong configuration. It's in a cis bond instead of being in a trans bond. Well, we would like to have it in position 2 and 3 in a trans bond. And it turns out this cell has an enzyme that does that beautifully. It's known as enoyl-CoA isomerase. And that enzyme does exactly what I just said. It converts a 3,4 cis into a 2,3 trans. Well, the cell knows how to handle a 2,3 trans because that's an intermediate in beta oxidation, so the cell proceeds with beta oxidation. Does that make sense? So that's the simple one. There's a more complicated one that I'm going to show you, but I'm not going to make you responsible for, okay? So the simple one is that this enoyl-CoA isomerase converts the cis 3,4 to a trans 2,3, and then beta oxidation simply proceeds. Yes, ma'am?
So what does the first enzyme do?
The first enzyme, this enzyme here? Well, this is just beta oxidation going on, cutting off 3 carbons at a time that you've already seen. So you have the dehydrogenation, the hydration, the oxidation, then the thiolytic cleavage. Make sense? Okay. So that's happening 3 times for this guy to get down to this point. Okay. All right. So we can handle the unsaturated fatty acids that way. This one I'm just going to show you, and you can just sit back and relax because I'm not going to hold you responsible for it. But you can see a little bit of the complexity of what the cell has to go through to oxidize more complicated unsaturated fatty acids. Let's say I've got something like this guy here. I go through, cut off my pieces, and all of a sudden I get down to my 1, 2, 3, 4 with a cis. The enzyme I just told you is going to fix that guy, right? So you're going to fix that guy. You're going to make a 2,3 trans, and everything's fine and dandy, and it cuts that off. You get down here, and after you've done that, what do you have? You have something that looks like this. 1, 2, 3, 4, 5. Okay? And so at 4, 5, it turns out to be more of a problem. The beta oxidation process starts, that is, we have the dehydrogenation to make the trans intermediate, but the enzyme that would normally handle the next step in beta oxidation doesn't like that double bond. And because it doesn't like that double bond, it actually uses another enzyme, I won't say it here so you won't be responsible for it, to convert these 2 double bonds into 1 double bond and then goes on. Okay? It's a little bit more complicated of a process, and for our purposes, I don't think it's necessary that you know that, but I just wanted you to see it so that you understand that there's more to unsaturated fatty acid oxidation than just that one enzyme. Okay. Well, the last thing I want to talk about with respect to fatty acid breakdown actually relates to something that the body does when it's running out of glucose. Okay? So I've said that if you are on a starvation diet, okay, for example, what's going to happen to you? Well, one of the things that's going to happen to you is you can exhaust your body's supply of glucose pretty readily. Or you might be diabetic and have some unusual aspects of your glucose metabolism. Or maybe for whatever reason your liver isn't putting out glucose at the level it should. Any of those can be dangerous situations. And the body typically has very immediate needs for glucose. So how does it deal with those immediate needs to provide glucose for the body? Well, it turns out that there's an alternate if there's no glucose available. I said that you can oxidize, you can take proteins, take the fatty acids, take the amino acids, convert them into oxaloacetate and make glucose out of that. But that takes a while. What if you have something, your blood glucose level is very, very low? What can you do? Well, one of the things the body does is it says, okay, we're not going to work with glucose. We're going to work with what we can use with fatty acids. And we're going to make ketone bodies. Okay? So ketone bodies are the body's approach to low blood glucose levels. They're a body's approach to low blood glucose levels. So let's look at how these guys get made in the body. And it turns out that ketone bodies get made by a reversal of the last step of beta oxidation. It's a reversal of the last step of beta oxidation. If we imagine that beta oxidation going all the way down until we have 4 carbons linked to a CoA, and we do beta oxidation on that, we're going to make 2 acetyl-CoAs, right? What's the enzyme that breaks the bond between the 2 carbons? Thiolase, okay? Thiolase, in this case, catalyzes the reverse reaction. Instead of breaking a bond between 2 carbons to make 2 acetyl-CoAs, it takes 2 acetyl-CoAs and puts them together to make that 4-carbon intermediate. You don't need to know the name. Okay? Then it takes a 3rd acetyl-CoA and makes something called HMG-CoA. This is a long name, but the abbreviation is HMG-CoA. This guy now has 6 carbons, and it's got a CoA molecule on there. Okay? This guy right here is made by an enzyme that we're going to talk about later. It's called HMG. HMG. Let me back up. I'm sorry. I'm not going to talk about this enzyme. I'm going to talk about this molecule. The HMG-CoA, we will see, is an intermediate in the synthesis of cholesterol. It's also an intermediate in the synthesis of ketone bodies. Now, when we go to make cholesterol, this guy shoots off another direction to make a great big ringed structure. But for the purpose of making ketone bodies, there's really only one more thing that has to happen. And the one thing that has to happen is an acetyl-CoA has to be removed from this, and when that happens, this guy right here is made. And you should know the name of that. It's known as acetoacetate. Yes, sir, Andrew.
So when you have your 4-carbon sugar,
Yes.
you're adding
This isn't a sugar. This is.
4-carbon
Yep.
molecule, you're adding CoA to it.
Yep.
You're taking that CoA off.
Well, yeah, it's taking it off in a different way than it went on, so it's actually making a different molecule. Yeah. So it's a little rearrangement it's doing. But suffice it to say that this is not what this was. You see, this is acetoacetyl-CoA, and this is acetoacetate. So they are different. Okay. Okay. Now, the important thing here is that this guy, 2 things, is chemically unstable. This is a ketone body. Acetoacetate is a ketone body, and it's chemically unstable. What does that mean? Well, it means that, for example, it can all by itself decarboxylate and become acetone. Acetone. Okay? Why is that significant? Well, first of all, acetone isn't very useful in your body. You don't really have a good way of metabolizing acetone. So it's a waste if this happens. And the other thing is that this can be, it's typically exhaled in a person's breath. You can smell it, okay? A person who is having problems with blood sugar You can actually smell it in many cases on their breath, and in many cases people don't know they're having a problem because this thing works pretty efficiently. It kicks in for you. One of the ways people learn that they're diabetic is by somebody smelling acetone on their breath. If you have a friend and you ever smell acetone on their breath, have them talk to their doctor if they don't know anything about diabetes, okay? Because this is an indication that there's something that's not going well with their glucose, with their sugar metabolism, okay? All right, well, I've said all that. What's this guy over here? This guy over here is the body's protection against that happening. Look at this. This is acetoacetate, which has a ketone right here. And down here it's been reduced to a hydroxyl. This is an NADH requiring reaction. This makes something called beta-hydroxybutyrate, and yes, you should know that. And beta-hydroxybutyrate, even though people call it a ketone body, it's not really a ketone body. Okay? But it's part of ketone body metabolism, because I'm going to show you in a second what happens with all this. Why have we gone through all this baloney? Is it just to learn a pathway? No, the body is doing this for a purpose. So everybody understands why this is made in the first place, right? This makes it chemically stable. This means it can travel in the bloodstream fairly readily. The more of this stuff there is, the more of this stuff there will be, and the more acetone you're going to smell in your breath, and the less efficient this overall process will be. Okay, well, let's imagine that the body has gone through this whole gyration, happening in the liver for the most part, making this guy right here. Okay? Now, the aim is that the body says, I'm running out of glucose. The reason I'm making this is to provide energy for the body. Particularly the brain is the number one thing it's making glucose, it's making energy for. Okay? This travels in the bloodstream. It gets to the brain. And what happens? The reversal of this whole process. The reversal of this whole process happens in the brain cell. What's the brain cell getting as a result of this? It's getting 2 acetyl-CoAs. It just got energy delivered to it. This will cross the blood-brain barrier. 2 acetyl-CoAs can be used in the citric acid cycle to make ATP. And bingo, the brain has what it needs. So ketone body metabolism is very important for delivering acetyl-CoA to cells that need it. Your brain is one example. There are others. Questions on that? Yes? Are there any instances where this goes wrong? You can look at almost any metabolic process, and there's almost always a genetic disease that will arise from some of that. So the answer is yes, probably. I don't know of anything specific to this, no. But I'm sure there probably are people who have issues with that. Yes, Anna. Okay, so ketone body metabolism, ketoacidosis, all these sorts of things relate to what's on here. It's a little bit more than I can really go into in this kind of a lecture. But it happens, again, because of low blood glucose. People who go on the ketone diet or they go on to low carbohydrate diet and so forth will be more susceptible to issues arising from this type of metabolism. Clear as mud? Okay. That's ketone body metabolism. Now we've got to talk about synthesizing fatty acids, because we've broken them down. The body has to not only break things down, but the body has to make things as well. Fatty acid synthesis does not occur in the mitochondrion. Okay? It occurs mostly in the cytoplasm. Well, this makes sense by separating the locations where a catabolic and an anabolic process occur. The cell doesn't have to worry as much about controlling them like it had to do the reciprocal regulation for glycolysis and gluconeogenesis. Okay? But the cell has got to get stuff out of the mitochondrion and out into the cytoplasm, because the starting material for synthesizing fatty acids is acetyl-CoA. And where is the cell getting acetyl-CoA from? Well, one of the places it's getting it from is right in here, because that's how we're breaking down fatty acids to make acetyl-CoA. We have to take them back out here to make fatty acids. Okay. Well, how does this happen? It turns out the cell uses a very cool trick to make this happen. Okay? Let's imagine I'm a cell and I'm going through the citric acid cycle, and I'm part of, let's say, a muscle cell. And the muscle cell is going through doing its things. And, of course, when the muscle cell is doing its things, what's happening? You're burning up that ATP. The citric acid cycle is going like crazy. When I take a rest and I'm not doing that exercising, what's happening? Well, the ATP levels will build up. The ADP levels will fall. And when the ADP levels fall, you know what happens ultimately to the citric acid cycle. What? It stops. So when the citric acid cycle stops, what's going to happen? Well, the first thing that's going to happen is that citrate is going to accumulate, right? The formation of citrate is a very energetically favorable reaction. We talked about that before. So citrate's going to accumulate. It can't be broken down further because there's no NAD. So citrate starts to pile up inside the cell. When citrate starts to pile up inside the cell, that is a clue to the in the mitochondrion. When it starts to accumulate inside the mitochondrion, it's a clue that we need to do something about it. This guy isn't burning all the ATP that we've got. Citric acid cycle isn't running. So there's a transport system that moves citrate out of the mitochondrion. It moves it out of the mitochondrion and moves it into the cytoplasm. Why is that important? That's important because the citrate in the cytoplasm can be broken down into acetyl-CoA and oxaloacetate. Now, that's the two things that came together to make citrate in the citric acid cycle. Now we're just breaking off an acetyl-CoA. The oxaloacetate gets dumped back into the mitochondrion, and the acetyl-CoA is here for the cell to use. Yes, sir?
Does it take a lot of energy to put them back together?
Does it take a lot of energy to put these two back together? It takes no energy to put them back together at all. That's how we started the citric acid cycle, and that had a very high energy bond arising from the CoA. And that was what was used to drive the process. So no, there's no energy necessary to put them back together. Yes, ma'am?
So when the mitochondria puts out the citrate into the cytoplasm, what happens to them?
So mitochondrion puts the citrate out into the cytoplasm, and then the citrate gets broken down into acetyl-CoA and oxaloacetate. So it's the reversal of the citric acid cycle reaction. The net result is this oxaloacetate gets pushed back into here, and the acetyl-CoA is left. This is what we call, what I like to call the citrate shuttle. Its only purpose is to carry acetyl-CoA out into the cytoplasm. And that's how acetyl-CoA gets into the cytoplasm for fatty acid synthesis. Other questions? Okay. Now, I'm going to show you in a little bit that the four reactions that you saw for fatty acid synthesis are reversed in fatty acid synthesis. Fatty acid oxidation going into fatty acid synthesis. Okay? So they're just basically reversed. Okay? But to get to that point, the cell goes through a couple of gyrations, and they also are a little bit odd. Let's talk about the first one. The first one is this guy right here. Okay? This guy right here, acetyl-CoA, which has 2 carbons, is made into malonyl-CoA that has 3 carbons. There's a bicarbonate, which is a carbon dioxide in water, is all it is. Carboxyl group's getting added. Now, this reaction takes energy. You can see ATP, but remember, this is happening when the cell has a lot of ATP sitting around, right? That's why the citric acid cycle stopped. So there's plenty of ATP for this reaction. This reaction occurs. This reaction is catalyzed by one enzyme whose name you need to know. It's known as acetyl-CoA carboxylase. Acetyl-CoA carboxylase. Now, acetyl-CoA carboxylase is the only enzyme that regulates fatty acid synthesis. It's the only enzyme that regulates fatty acid synthesis. If I could inhibit this enzyme, I probably wouldn't have as much fat sitting around, right? This enzyme is activated allosterically by citrate. And guess what's been pouring out of the mitochondrion? Citrate. So citrate activates this enzyme, and the end product of fatty acid synthesis inhibits it. That's palmitate, palmitic acid. So this enzyme is feedback inhibited. Remember, feedback inhibition occurs when an end product of a pathway inhibits the first enzyme in the process. So palmitic acid inhibits this enzyme. Yes, Tessa? Palmitic acid is not on here. I haven't shown you the entire pathway yet. So this is just the first step. Okay? But palmitic acid is the last product of the pathway. Okay? Okay. Everybody following with me so far? So what have we done? We've taken a 2-carbon molecule, we've made a 3-carbon molecule, and the odd thing is only 2 of these 3 carbons are going to make it into a growing fatty acid chain. Only 2 carbons are going to make it into a growing fatty acid chain. Let's see how that happens. Oh, there's the acetyl-CoA carboxylase reaction. You've already seen it, so I'm not going to show you that again. Okay? How do we synthesize palmitic acid? Here's the reaction that you've, actually, one other thing I need to tell you. One other important thing. Okay? CoA is the carrier molecule for oxidation. The synthesis swaps out that CoA and replaces it with something called ACP. So CoA is replaced by it's actually a protein called ACP. If you're curious, it stands for acyl carrier protein. Yes, sir? It's swapped out as a very early step in the process. So we're going to see that synthesizing a fatty acid requires 2 things to start. It requires that malonyl-CoA that I just described, and it's going to require an acetyl ACP. Okay, everybody with me? Here is our acetyl ACP, okay? And to be honest, I don't even know what that KSA thing is there. I think, don't worry about it for the moment, okay? We've got acetyl ACP, and we've got malonyl ACP. There's the malonyl group. We've taken malonyl-CoA, and we've replaced the CoA with an ACP. Now, the important thing that happens here is that 2 of these carbons are going to join with 2 of these carbons to make a 4-carbon molecule. So I'm starting with something with 2 and 3, and I'm going to end up with something with 4. That means at some point I had to lose a carbon dioxide. The carbon dioxide that I lose is the one that I put on. I put it on, and I take it off. Okay? So 2 carbons plus 3 carbons. Okay? And this should just say ACP. I don't even know why I've got that on there, but that's just ACP right there. Malonyl ACP. I'm sorry, acetyl ACP right there. All right. Malonyl ACP, acetyl ACP. We make a 4-carbon intermediate. Now, every other step is the reversal of what you learned in beta oxidation. Okay? There's a ketone, gets reduced to a hydroxyl. Instead of being oxidized, it's getting reduced. This guy right here loses a water instead of this reaction, which is a gain of water. By losing water, a double bond is formed. And that double bond then gains 2 hydrogens through a hydrogenation instead of the reverse reaction, which is a dehydrogenation. Consequently, we now have 4 carbons. We've reduced the ketone to a CH2. Okay? This process is then repeated. Another malonyl ACP will be added to this so that we gain something that will have 6 carbons. And we keep repeating that over and over and over until we get to 16 carbons. That's palmitic acid. By the way, don't worry about the enzyme names. I'm going to give you a real simple way to know all the enzyme names there. Okay? Don't worry about them. In fact, I'll tell you right here. You need to know the acetyl-CoA carboxylase name. I told you that one, right? All these other enzymes that we see right here turn out to be all part of a giant complex. In the giant complex, we give the name fatty acid synthase. Okay? So all the other reactions are known as be catalyzed by fatty acid synthase. Fatty acid synthase can only make fatty acids up to 16 carbons. That's why we stop at palmitate. So we get all the way up to 16 carbons, and when we get to 16 carbons, the 16 carbon piece is released as palmitoyl-CoA. Palmitoyl ACP, and then the ACP is replaced by a CoA. So at the very end of the process, the ACP is taken off and replaced by a CoA. Questions? Yeah, it's a good question. Does the body ever stop before it gets to palmitic acid? As far as I know, it goes all the way. I don't know if it's stopping. Now, point out a couple things to you. There's a couple reductions in here. The reductions, this guy going to the hydroxyl, for example, the electrons come from NADPH. It's the first time you've seen that. Same thing down here. NADPH are the source of electrons. Okay? One other difference, this guy is in the D configuration, and the corresponding molecule in the oxidation was in the L configuration. Okay? So in the synthesis process, that hydroxyl is in the D instead of being in the L configuration as it is for oxidation. This is interesting to look at. This is the structure of coenzyme A, CoA. This is the structure of the end of ACP. The handles are identical. Okay? This one's just attached to a protein. This one's just attached to an AMP. That's the only difference. Okay. What do you guys say to finishing a little early? Everybody to life. Okay. Let us sing a song, and then we'll call it a day. How's that? I put everybody in a good mood with that one. Okay. Should do this more often. Okay, please sing loudly. The 16-carbon fatty acid palmitate gets all the carbons that it needs from acetate, which citric acid helps release from mitochondria matrices. Oh, a shuttle's great when acids are synthesized. Carboxylase takes substrate and it puts within dioxycarbon carried on a biotin. CoAs all gain a quick release replaced by larger ACPs, and it all begins when acids are synthesized. A malonate contributes to the growing chain. 2 carbons 7 times around again. For saturated acylates, there's lots of NADPH that you must obtain when acids are synthesized. Palmitic acid made this way all gets released. Saturases act to make omega-3s. The finished products, big and small, form esters with a glycerol, so you get obese when acids are synthesized. Okay, so I'm going back to my office if you guys would like to follow me back there, meet me back there, whatever. I'll be there to go through stuff with you.